Today we're going to look at cells, and cells are going to be the smallest living unit. So for those of you in Biology 241, this is the first time you're getting to actually look at something alive in class. So the cell is the fundamental unit of life. So basically all living things are made of cells, and all cells come from other cells. Cells are the building blocks. It's the basic unit of structure and function. And in a human adult, we usually have about 75 trillion cells. So human cells are going to fall into two basic classes. Your sex cells or germ cells are your reproductive cells. They're sometimes called your gametes. They're going to include the sperm in the male and the eggs or oocytes in the female. All of your other cells are somatic cells. Soma means body, so this is going to be everything in your body except the sperm and egg. Cells are small, really small. So to give you kind of an idea here, this shows the human egg cell, which is generally considered to be the largest human cell. Red blood cells are extremely small here, so looking at their relative size. And then muscle cells, it's not that they're overall large, but they're long and skinny. So here's an example of a smooth muscle cell. So we start out as one zygote cell from a few sperm and egg, and that will differentiate into over 200 different cell types. These are just a few of the cell types here you can see in these different tissues. So not all cells are the same, but they are going to share some general structures and characteristics. So there's three main regions to the cell: the nucleus, the cytoplasm, and the plasma membrane. So the cytoplasm is this area between the nucleus and the plasma membrane. So if we look at a typical human cell, some of the things that you're going to have: your chromatin in the center, this is your genetic information. The nucleus is surrounded by a nuclear envelope. You're going to have the cell membrane on the outside, ribosomes, your smooth endoplasmic reticulum and rough endoplasmic reticulum, depending on whether or not it has ribosomes on the surface, mitochondria and centrioles, and basal bodies. Microtubules can make things like the flagellum. You can have microvilli, secretory vesicles, your Golgi apparatus, microtubules, lysosomes, and then your microtubules can also make up the cilia. So these are some different things that you would see in a typical human cell. Your plasma membrane is sometimes referred to the cell membrane. It's your outer boundary of the cell. This is its boundary to the outside world. It's made up of a phospholipid bilayer, so it's two layers of phospholipid. This is going to give it some unique properties in that it's semi-permeable and selectively permeable. Some things are going to be able to pass through, others are not. It's going to help regulate what goes in and out of the cell. So some molecules will pass through freely, others are going to require some assistance to get across. So the phospholipids are going to create the bilayer. It's going to have a polar and nonpolar end. You'll have glycolipids that can have protective functions and help to identify cells. So different cell types will have different glycolipids on there. The cholesterol is bulky. It's going to help to reduce the permeability. It's also going to play an important role on acting as a buffer to keep the cell from being too soft and too liquid, or having the membrane get too hard and not be fluid at all. So we can also have glycoproteins. They can be transmembrane or integral. So they may span the entire membrane. They can be peripheral, where they will be embedded on either the cytoplasmic side or the extracellular side. And then you can have glycoproteins and glycolipids that help in cell-to-cell recognition. And the glycocalyx. These markers on the cell are very important when we look at organ transplants. So we say that it's a fluid mosaic model because these things are always moving around in the cell membrane. So things that can permeate the membrane easily: oxygen, carbon dioxide, other small nonpolar molecules, some water molecules. Things that are not going to go across would be glucose, Other large polar water-soluble molecules, ions, they're not going to go. And in some cases, water molecules don't cross easily. So this is a nice picture here. It's showing your phospholipid bilayer here, the polar head. It's going to be hydrophilic. It's going to like water. You've got the fatty acid tails that are going to be hydrophobic. This is going to be the cholesterol. This is going to prevent these from packing too densely together. So here you can have glycolipid on the cell. You can have these pores or channel proteins. Channel proteins can be an integral or transmembrane protein. This can allow specific things to pass. You know, glycoproteins on the outside. And then you have your fibers underneath that are part of the cytoskeleton. So channel proteins, these will create a transient hydrophilic channel. This will be more inviting for the small molecules and ions to flow in and out of the cell. Your carrier proteins, these will selectively interact with small molecules or ions. They're going to be picky. They're going to be selective on who they're going to allow to cross. Cell recognition proteins, these are important for the cell's identity. There's individual specific groups of proteins on the extracellular side of the membrane. So your MHC, your major histocompatibility complex, or the HLA, human-like leukocyte antigen, is important to match for organ and tissue donors to avoid rejection of the transplanted organ or tissue. They are going to be different unless your person is a genetically identical clone to you. However, you want to limit the differences in there to limit the type of reaction. Receptor proteins interact with specific molecules to transmit some type of signal or communication. So these can range from electrical to chemical or physical contact. You can have hormone receptors. Enzymatic proteins, these will act as catalysts and speed things up for some specific reaction that's going to lead to a cellular response. And then your CAMs, or cell adhesion molecules, will guide interactions between cells. So when we look at membrane proteins and functions, there's over 50 different kinds of proteins so far that have been found in the plasma membrane of red blood cells alone. So this just kind of summarizes here. Integral proteins will penetrate into the hydrophobic core of the lipid bilayer. A transmembrane protein is going to be an integral protein that spans the entire membrane. Peripheral proteins, they're not embedded, they're appendages bound to the surface, and they can be exposed to parts of integral proteins. And then the integrins are integral proteins that will attach to fibers of the extracellular matrix. Excuse me. Here's some additional pictures of types of membrane proteins. So an ion channel here is an integral protein. It's going to allow some of the specific ions to move through a water-filled pore. A carrier protein here that's an integral protein, it's going to carry a specific substance across. It does this by changing shape. So the carrier proteins are also known as transporters. The receptor here is another integral protein. It recognizes a specific ligand that's going to alter the cell's function in some way. So then we can have enzymes. They can be either integral or peripheral, catalyzing a reaction on the inside or outside. Linkers, they're integral and peripheral. They can anchor filaments inside or outside the plasma membrane. These help with stability. And then the MHC, which is a cell identity marker. So when we look at things crossing the plasma membrane, passive processes are ones that are going to cross the cell membrane without requiring energy. They're going to rely on the kinetic energy of the individual molecules or ions, where active processes the cell is going to have to use energy. This is primarily going to come from breaking down ATP to move a substance across the membrane. So with these, it's generally going to move against the concentration gradient. Passive are usually going to move with the concentration gradient. So diffusion looks at the movement of solutes. The rate of diffusion is going to depend on the steepness of the concentration gradient, the temperature, the mass of the diffusing substance, the surface area, and the diffusion distance. So when something moves directly through the lipid bilayer, we call it simple diffusion. A lot of times they're going to need the help of a membrane protein. So we call this facilitated diffusion. Facilitated means it has a helper. The rate's going to depend on the concentration gradient, temperature, mass of the diffusing ion or molecule, the membrane surface area, and the diffusion distance again. So here's an example. We have a concentration gradient that is higher here in this extracellular fluid and lower in the cytosol. So oxygen and carbon dioxide, they can do simple diffusion. Potassium, it's going to move as an ion in most cases. It's going to need a channel-mediated protein to help it get across. It will be facilitated diffusion. With glucose, we can have carrier-mediated facilitated diffusion. So osmosis is going to look at the movement of water. The net movement of water through a selectively permeable membrane from an area of higher concentration of water or lower concentration of solutes to one of lower concentration of water. The water wants to spread out. You can pass through the plasma membrane either through the lipid bilayer by simple diffusion, or it can use aquaporins, which are integral proteins or channel proteins that are specific to allow water to cross. So here we can look at this, where you've got this selectively permeable membrane. These solutes are too big to cross. So what's going to happen is the water is going to move over here to try and dilute this. So when it reaches equilibrium, now this side is dilute. So when it's doing this and you don't allow it to move, there's something on here blocking it, we've created osmotic pressure. So tonicity is the strength or solute concentration of a solution in relation to osmosis. So in a cell, the solute concentration of a solution with respect to that solute concentration inside the cell. So when you have something that's isotonic, the net solute concentration of the solution is the same as what's inside the cell. If it's hypotonic, the net solute concentration of the solution is less than what's inside the cell. So in an animal cell, what's going to happen is it's going to move into the cell and eventually you'll have it burst. So if it's a red cell, we call it hemolysis or hemolysis. If it's hypertonic, the net solute concentration of the solution is greater than that inside the cell. So for an animal cell, it's going to pull the water out and the cell will shrink. We refer to this as crenation. So this is showing it here with the red blood cell. In an isotonic solution, it's happy. This is what we are aiming for when we give a person an IV. We don't want to have a plain water IV and have the water rush into the red blood cell and cause it to burst. Likewise, we don't put it in an extremely salty solution because it would cause the water to be pulled out and the cell to dry out. So this will be how filtration occurs a lot in cells. You have a pressure gradient that's going to push the solute-containing fluid or filtrate from the area of high pressure to low pressure. It's going to force the water and solutes through the membrane or capillary wall using hydrostatic pressure. So with active transport, you move against the concentration gradient. So your solutes get transported across the membrane. It's going to require the use of ATP for energy. It's going to go to an area of lower concentration to higher concentration. Your sodium-potassium pump is an example of this. So while you have a high amount of potassium inside the cell and a high amount of sodium outside, what will happen is it's going to pump 3 sodiums outside, 2 potassiums in, to increase those concentration differences even more. So moving things in vesicles. A vesicle is a small spherical sac. It's formed by budding off from a membrane. It's just a membrane-bound substance. So when a cell does endocytosis, the materials go to move into a cell. What will happen is the cell basically engulfs or wraps the membrane around that substance to take it in. So there's 3 types: receptor-mediated endocytosis, phagocytosis, and bulk phase endocytosis or pinocytosis. So with exocytosis, it's the reverse. The vesicle is going to fuse with the plasma membrane, and when it does, it's going to release the contents that were in that vesicle out into the extracellular fluid. So transcytosis is a combination of endocytosis and exocytosis. So here we're looking at phagocytosis. You basically wrap the cell membrane around this little particle, in this case a microbe. It's going to engulf it, doing phagocytosis. The pseudopodia wrap around it. It takes it in. You have a lysosome fuse with it. It's going to dump the enzymes in there and digest it. So this is what white blood cells will do. So with bulk phase endocytosis, you're going to wait for multiple of these little particles to come in and form the vesicle. And then you can have the lysosome fuse with it to have the lysosomal enzymes dumped in and digestion to occur. So the cytoplasm is going to be between the plasma membrane and the nucleus. It's going to have intracellular fluid in there that surrounds the organelles. A lot of your chemical reactions take place. A lot of times energy is going to be released by these reactions, and then they provide building blocks to do other things in the cell. Maintenance, build structures, have the cell grow, carry out normal functions. The organelles are specialized structures within the cell. It sounds like mini organs because that's what they act like. This is a lot of your metabolic machinery. The cytoskeleton, it literally means cyto is cell, and skeleton is a protein filament, that set of protein filaments that are going to go throughout the cytosol. It provides support. They'll include the microfilaments, intermediate filaments, and microtubules. And then we can have inclusions, which are non-functional chemical substances. This can be granules of glycogen, pigments, liquid droplets, vacuoles, or crystals. So when we look at the cytoplasm, you've got the cytosol portion that's a semi-gelatinous intracellular fluid. It's going to suspend the organelles. You'll have ions, nutrients, waste, enzymes in there in the inclusions. And then you'll have the organelles that are going to perform your specialized tasks. Excuse me. Non-membranous organelles would be the cytoskeleton, cilia, centrioles, ribosomes, proteasomes, microvilli. Then some are going to be membrane-bound: mitochondria, peroxisomes, lysosomes, proteasomes, endoplasmic reticulum, nucleus, Golgi bodies, and vesicles. So this is just another illustration of a cell here. You can see different parts of it. So the cytoskeleton is going to be composed of protein filaments that are going to go throughout the cytosol. They help to determine the cell's shape, move the organelles, create and move membrane vesicles, basically acting like little railroad tracks, organizing the contents of the cell, and then moving the chromosomes during cell division. So some of them are going to be smaller, your microfilaments, some are larger, microtubules, and then some are medium-sized, your intermediate filaments. So the centrosome is located near the nucleus, and it consists of 2 centrioles, and then the pericentriolar material and the mitotic spindle. Your cilia, which you can see here, are short little hair-like projections along the cell's surface. In human cells, they're going to move fluids along the cell's surface. So you have these in your trachea that help to move mucus up. Flagella are longer than cilia, and they move an entire cell. The tail of a sperm is an example of this. Cilia are going to tend to move things perpendicular. The sperm are going to tend to push things in a line forward. And then microvilli are little finger-like projections that are going to be on the cell. They're going to help increase the surface area. In humans, we see a lot of those in the digestive system. Ribosomes, they're the smallest and most numerous of the organelles. They're going to help to make your polypeptides and protein synthesis. There's two types. The free ribosomes are found in the cytoplasm. They're generally going to make proteins that are going to stay inside the cell. Then we also have some that are fixed or attached to the endoplasmic reticulum. They'll make your secretory proteins and cell membrane proteins. The endoplasmic reticulum is a membrane network inside the cell. Rough ER has ribosomes on the surface, more involved with protein synthesis, where the smooth ER does not have ribosomes on the surface. It's more involved with carbohydrate, steroid, and lipid synthesis. It is also important for drug detoxification. So one of the ways that a person will build a tolerance to a drug is they will proliferate more smooth ER in the liver cells. They can store calcium in muscle cells. We call it the sarcoplasmic reticulum there. So the Golgi apparatus is kind of like a UPS store or a FedEx store. It's going to complete modification of proteins from the rough ER. So what will happen is it will get taken to one side of the Golgi apparatus. It's going to be transported through and come out the other side and be packaged and have a label on it. So this can transport organic molecules into vesicles and help make them into lysosomes. So lysosome, lys means to split something open, some is a body. These are little digestive bags or garbage disposal. They'll have acid hydrolases and various other digestive enzymes in there. This will help break down food particles, recycle old parts of cells, worn out cell parts. We have them in phagocytes and they help to destroy invaders. Peroxisomes are going to have enzymes for oxidizing certain organic molecules with the release of hydrogen peroxide. So it's toxic, but it will break down into water and oxygen. So this is another example of an animal cell here. You'll notice that these are all artist representations of cells. When you actually look at a cell underneath a microscope, you cannot see most of these things in there. So more pictures of your cytoplasmic organelles. So this is showing exocytosis happen, where this is going to fuse with the membrane and spew the contents out. So mitochondria, these are involved with cellular respiration. The more of these a cell has, the more energy it can produce. So sometimes they're referred to as the powerhouse of the cell or energy transformers. They're going to be doing the conversion of chemical bonds to the usable form of ATP. ATP is like your energy currency. So the number of mitochondria you have depends on the cell's function. Heart cells, sperm cells, and some of your muscle cells have high energy requirements and will have a lot of ATP, or have a lot of mitochondria to make that ATP. So this is showing here the cristae are the folds, and the matrix is going to be the material between this outer membrane and the folds. The nucleus is the control center. It's going to contain your genetic information, or DNA. There's 3 regions to it. The nuclear membrane that's on the outside, the nucleolus, which is going to assemble the ribosomes, your chromatin, which is your genetic information. Chromatin just refers to the loosely coiled genetic information inside the nucleus. The gene is a region of that DNA that makes up that chromatin that's going to code for a particular polypeptide or RNA. So as humans, our genome is going to consist of 23 pairs of chromosomes. Each chromosome is going to contain DNA, and it gets combined with histone proteins to form chromatin. So what you can see here is you've got this DNA and these histone proteins, and the DNA wraps around it. When it does, it makes these little beads we call the nucleosomes. So the histones allow the DNA to be tightly packed. It will continue to do this packaging, and eventually it loops and coils itself into the chromosomes. So the two legs of the chromosomes are the chromatids, sometimes called sister chromatids. So after DNA replication, the sister chromatids are connected at the centromere, here in the center. So these are your nucleosome beads that are the DNA wrapped around the histones. When we look at the cell's life cycle, it's got two major periods. You've got interphase, where the cell is going to grow. It's going to carry on its metabolic processes. It's doing its main life's purpose here, whatever the region is for, whatever that is for the cell. And then during cell division, we call it the mitosis or mitotic phase. Here the cell replicates itself. This will produce more cells for growth and repair. So it's a little hard to see on here, but it's estimated that interphase in most cells is about 90% of its lifetime. So we can divide up interphase. You can have the G1 or cell growth phase. S phase here, it's going to synthesize its genetic material, basically making a copy of it for when it goes through mitosis. And the G2 phase where it's going to grow some more, and then it will go into mitosis, which will have 4 subphases to it. So in interphase, GO phase is what will happen when it's a non-dividing cell. This is actually quite a few of your human cells will be here. Sometimes they can be called back out of this phase, but some cells like nerve cells rarely leave it. G1 is the first gap. Here it's doing its normal metabolism, normal cell reactions, and its growth. S phase, you're doing the DNA synthesis, getting ready to be able to divide. G2 phase, or the second gap. Here you're going to have growth and preparation for the big day of division. So mitosis is the division of the nucleus, sometimes called karyokinesis. So we've got 4 phases to that: prophase, metaphase, anaphase, and telophase. And then cytokinesis is the division of the cytoplasm. So when we go to divide a cell, we need to make copies of the DNA. We have to do some DNA replication. So this genetic material gets duplicated so the cell can have 2 copies for 2 cells. So you start out with your main strand. It will separate, and one of them will be a template strand, and then it will fill in with its complementary partner to make the newly synthesized strand. So we refer to this as being semi-conservative replication because these daughter strands here are going to be half old strand and half new strand. So this will occur towards the end of interphase. So here you're going to have chromatin. It's protein and DNA. When it's uncondensed, it's chromosomes when it's condensed. This here also illustrates your semi-conservative model. You have this parent molecule that's going to separate, and the 2 daughter molecules are going to be half parent, half new molecules. So that parent molecule will have 2 complementary strands. Each of those base pairs is paired by hydrogen bonding with its specific partner. So in DNA, it's going to be A and T, or adenine and thymine, and guanine and cytosine, G and C. So the first step in replication is to separate these 2 strands. The parental strand will serve as a template to determine what order we're going to put those nucleotides in the new complementary strand. Those complementary nucleotides are going to line up. They're going to connect their sugar phosphate backbones and pair up so that that new daughter DNA is half old, half new. So in interphase, you've got G1, S, and G2. So it's showing here the daughter cells doing their normal function. There's this restriction point here. If they get past that restriction point, then they will go on to divide. late interphase, the cell's past that restriction point. So it's going to replicate its DNA as well as centrioles, mitochondria, and make extra membrane to get ready for division. So mitosis is the division of the nucleus. It's going to result in the formation of two daughter nuclei, which then in human cells will undergo cytokinesis, where you'll divide the cytoplasm. It's usually going to start when the mitosis is almost done, and you end up with two new daughter cells. So these two identical daughter cells are going to be genetically identical or clones. So remember, interphase is that non-dividing phase. It's doing its normal metabolic activity. Prophase is going to be the first part of cell division. Here, your centromeres are going to migrate to the poles. During metaphase, you're going to have the spindle form the centromeres, and those will be attached to the chromosomes that are aligned along the center of the cell. In anaphase, the cell is going to begin to elongate. Those daughter chromosomes get pulled towards the poles, and in telophase, your daughter nuclei begin forming. So here you'll usually see the cleavage furrow for cell division. Exactly when you do cytokinesis can vary cell to cell. So here, looking at prophase, what you can see here is the chromosomes have started to condense and become visible. This nuclear envelope and the nucleolus are going to start to disappear, and you start to see the spindle apparatus here. So in metaphase, this has a distinct appearance underneath the microscope. Your sister chromatids or chromosomes are going to be lined up along the metaphase plate, which is in the equator of the cell. So in anaphase, the sister chromatids are going to separate. So now they are chromosomes. They're going to move towards the opposite poles of the spindle. They're going to be pulled by the spindle fibers, which are microtubules. So in telophase, those chromosomes reach the poles. You're going to start to make a new nuclear membrane around those chromosomes. The nucleoli are going to reform and the spindle disappears. And what you have are two daughter nuclei. So here you can see the cleavage furrow forming. This is when the cell is going to start to pinch in two. So you actually have little actin fibers in here. They're going to act like a drawstring to pinch this off so that you can have two new daughter cells. So this picture combines all of these into one if you're looking for a single picture to study this process. So this is showing a linear progression of this process. So these are showing some that are underneath the microscope. So prophase, some in metaphase, so this distinct appearance. Anaphase, this one they're separating, and in telophase. So meiosis is your sex cell production in here. We use this for sexual reproduction. You're going to create these unique cells called gametes, your sperm and egg. So a human somatic cell is going to have 46 chromosomes. They're diploid, so the chromosomes are paired. Gametes are haploid. They're only going to have one copy of the chromosome. So in humans, our haploid number is 23. So we'll have homologous chromosomes, which are going to be the paired sets of chromosomes that are going to have the pairing from both parents. They're not genetically identical. Sister chromatids are duplicate copies. They are genetically identical. So in the first division of the chromosomes, they're going to pair up and exchange DNA. We call this process crossing over. It helps give more genetic recombination. So you can have different regions of the chromosome swapping. So instead of passing an exact copy of mom's or an exact copy of dad's, what you're going to pass on is a mix of the two. So at fertilization, you'll have a male and female gamete combined to create a new diploid individual. So you take the male's haploid cell, the female's haploid cell, put them together, you get a diploid individual that will undergo mitosis. This here is showing where you've had some crossing over. This little part of the chromosome swaps places. So for meiosis, we can divide it into two main phases, Meiosis I and Meiosis II. In Meiosis I, you're going to have Prophase I, Metaphase I, Anaphase I, and Telophase I that is going to look very similar to mitosis. And then with Meiosis II, you have Prophase II, Metaphase II, Anaphase II, Telophase II. In this first one, you're going to copy the genetic material. In the second one, you do not copy it. Therefore, you end up with 4 cells that are haploid. So to kind of summarize what goes on in these two things, in mitosis and meiosis, you're going to have DNA replication occur during interphase. Mitosis only undergoes 1 division. Meiosis is going to undergo 2 divisions. There is no synapsis of homologous chromosomes in mitosis. You don't have homologous chromosomes. You only have 1 chromosome. So in meiosis, you're going to have that synapsis and the crossing over. That's going to occur between the non-sister chromatids. So the number of daughter cells you're going to get, in mitosis it's 2. They're genetically identical. In meiosis, there's 4. They're genetically different. So the role in the cell, cells from mitosis are going to do all of the various functions in the body. The ones from meiosis, they're gametes. They're going to be your sex cells, and their job is to increase genetic variability into the new organism. So 3 events are unique to meiosis. 1, the synapsis and crossing over, where you have the duplicate homologs pair up. They make the synaptonemal complex where they join, and you have crossing over. You have homologous pairs at the metaphase plate during meiosis I, and then you have the separation of the homologs where the duplicate chromosomes of the homologous pairs separate, but the sister chromatids are going to stay together. They do that because that enzyme that's going to cleave them will be released in 2 parts. Meiosis I is sometimes referred to as your reductional division because it halves the number of chromosome sets per cell, goes from diploid to haploid. Sometimes meiosis II is referred to as the equatorial division. Here the sister chromatids separate to make your haploid daughter cells. So the big reason for doing sexual reproduction is genetic variation. So the behavior of the chromosomes during meiosis and fertilization is going to give most of the variation. This is going to give the organism better survival rates because there's more options. If something goes wrong or there's a drastic change in the environment, with more options out there, you're more likely to have one that's going to survive. Part of the reason for this is we have independent assortment of chromosomes. So at metaphase I, your homologous pairs are going to have 1 maternal and 1 paternal chromosome. So each may pair with either a maternal or a paternal close to a given pole. You basically have a 50-50 chance that a particular cell will get the maternal chromosome or the paternal chromosome. And that will happen independently for every other pair of chromosomes. So for humans, you've got 2 possibilities, getting the maternal one or the paternal one. And you've got 23 haploid chromosomes, so that means when you produce your gametes, there is 8.4 million possible combinations of your chromosomes that can go into producing your sperm and egg. Crossing over, it's going to produce recombinant chromosomes from 2 different individuals. It's going to increase that number even more. This kind of allows you to see a little bit with the genetic variation. This is only looking at 2 chromosomes here. So you can produce offspring where both of them got the blue one or both got the red, or you can have where they each received one of the other. So protein synthesis. Proteins are an expression of the genetic information. Your genetic information is basically the instructions to make these proteins. Transcription is going to be the process where the DNA sequence is copied or transcribed, and it will make an RNA sequence. This is going to occur in the nucleus. Part of the reason for the cell doing this, that DNA is the master copy of instructions. You don't want to lose it or have it damaged. The RNA is almost like a photocopy. It's the working copy that you can use. So if that RNA contains a sequence that's meant to encode proteins, we call it a messenger RNA or mRNA. So this will be used to make new proteins in a process called translation. It's basically in a code, and the cell is going to translate the code to build it. The gene is the DNA segment that's going to have that blueprint for building a protein or polypeptide. So your start point is going to be the nucleotide where the RNA synthesis actually begins. It's usually going to be a promoter that's typically several dozen nucleotides upstream from the start. We have transcription factors in eukaryotes that are going to be collections of proteins. They're going to help to mediate the binding of the enzyme RNA polymerase on there. You need the RNA polymerase in order to make the RNA. So with this, you'll have the initiation of transcription. This only happens after the transcription factors attach to the RNA polymerase and binds. So the process is a little bit different in prokaryotes, which we are not going to worry about right now. So when you reach the end, you've got to have termination. The RNA polymerase II will transcribe this polyadenylation signal. It's basically this AAU-AAA in the pre-mRNA. And then 10 to 35 nucleotides downstream, it's going to cut free the pre-mRNA. So this picture shows it nicely here. You've got this region that's the promoter. The RNA polymerase is going to hop on here. This is your transcription unit. It's what it's going to read. So it's going to unwind the DNA and start to make this RNA transcript. It's going to be complementary to this DNA. It's going to continue along here. It opens that up enough to be able to read it, and then it closes back up. And you're going to make this RNA transcript. When it gets to the end, it pops off, and that RNA transcript is complete and released. We do have multiple types of RNA in the cell that play several different roles. Transfer RNAs or tRNAs, these are going to be ones involved with transferring the appropriate amino acid to the ribosome for building a protein. When you have your amino acids, they are going to have tRNAs attached to them. That's going to be what the ribosome looks at to see, does this one match up? Do I have the correct amino acid? The ribosomal RNA or rRNA is going to actually be part of the ribosomal structure. It helps form the part where proteins are built. Your messenger RNA or mRNA carries the instructions to build a protein, and this will carry it from the nucleus out to the ribosomes. So transcription occurs in the nucleus. It's going to transfer the information from the DNA's base sequence to make the complementary base sequence for the mRNA. Translation, this occurs in the cytoplasm. You have the base sequence of the nucleic acids. It's translating the code to make the amino acids. The amino acids are your building blocks of the proteins. So with translation, your ribosome is going to read these codons. Basically, you only have 4 base pairs. You've got 20 different amino acids. You need them to be in sets of 3 in order to have enough. So the ribosome will read them. You have these codon charts. Nobody memorizes these. So when you go to read this, say for example, you're looking at AUG, your start codon. First letter is A. You're going to find that along this side. Second letter, U. You find this up here. When you bring those two lines together, it will bring you to a box. You find the third letter over in that box and come across. Where all those lines intersect, that will tell you what your codon is. So AUG is the start codon. It's kind of like starting with a capital letter at the beginning of a sentence. That's what you look for to know you're at the start. When you're going to translate the genetic code, you always start with the first AUG. So we also have some stop codons here. So if you have UAA, for example, you go UA, that brings you to this box, A over here, that tells you it's a stop. So there are 3 stop codons, UAA, UAG, UGA. This is like having a period, exclamation point, or a question mark at the end of a sentence. It gives you the sign that you're at the end. So this is showing transcription occurring here. This is similar to copying the DNA, but instead of making a second DNA strand, you're going to make an RNA strand. So you've got the promoter, the gene, the terminator. The RNA polymerase is going to be here. What it's going to do is read these codons, 3 base pairs at a time. You're reading these base triplets, and it's going to tell you which tRNA to incorporate. So in translation, you use the mRNA and the ribosomes to make your proteins. The ribosome comes as a large and a small subunit. Some books are going to draw them to look more like a hamburger bun model, so when you look in your textbook, it will probably look a little different than this picture. So you've got this mRNA here, and then you're going to have these sites. You have an A site where you're going to bring in or accept the new polypeptide. The P site's going to be your growing polypeptide, and when it gets down here, there's an E site that's going to eject it out. So this here is going to show basically you've got your start codon. It's going to read that. It will move on. It's going to bring in the new one. Moves into the P site. You make your polypeptide chain, and then you can repeat the process going through and through. So I will include a couple of animations of this. I would encourage you to look at transcription and translation animations online, looking at various ones until it starts to click and make sense. The animations work a whole lot better than the diagrams. So this is trying to summarize the process again. Here you've got your DNA that you've made this mRNA from. The mRNA is going to leave the nucleus, attached to the ribosome out in the cytoplasm. It's going to use the tRNA to help bring the correct amino acids in. It's reading the codons along here. You're going to form this polypeptide chain, and you will eventually release it, and it will spontaneously fold up into your protein.